Good afternoon. My name is Katherine Beaverson, and I am the Vice President of Global Patient Advocacy with Dyne Therapeutics.
And my name is Wes Miller. I'm a Chief Medical Officer at Epic Bio.
That's great. So each of our panelists have their own microphone, so that's super. There's a whole series of questions that we have included here, but I wanted to start at the bottom, honestly, because I wanted to ask some of the questions around how the patient community can help accelerate clinical trials and ensure that they're well designed, which are maybe two slightly different questions. So we'll start with that and with Wes, since we already had some words from Avidity. Is how could the patient community get involved, accelerate, and make sure we've got well-designed trials?
Yeah, sure. So first of all, so that you contextualize my opinion, my background is that I'm a physician, I'm a pediatrician, hematologist, and oncologist, did blood stem cell transplantation for children with various diseases for over a decade, have been over in the industry side for about 7 years now in various small biotech companies, exclusively developing therapies for people with rare diseases using gene therapy or gene editing technology. Epic Bio is our very first program that we have and that we expect to be in the clinic is for FSHD. And so, we're somewhat of, I think, a newcomer to the space. I would say that in general, as a physician who's been in clinical trials and trying to develop therapies for rare disease, I think FSHD could use high quality universal biomarkers of disease. I think that's probably a self-evident statement, but I mean what I mean by that is not only understanding ways to understand in a moment in time how severe the disease is, or that might predict the disease and how it's going to progress, so-called prognostic biomarkers or severity biomarkers, but also therapeutic response biomarkers, meaning if we measure something and we measure it later, if it changes, or maybe if it stays the same, does that then predict that that intervention or treatment or therapy is going to translate to clinical benefit? So with that being said, it feels to me that there is a lot of movement in FSHD in trying to identify biomarkers. And sometimes I have to check myself. I think of a biomarker as something that's only measured from the blood. That's not true. Many biomarkers are measured from the blood, but we can also measure biomarkers in muscle. We can measure biomarkers on imaging, on MRIs or ultrasounds. And so I would say that one thing is really trying to promote, participate in, and advance studies of understanding these biomarkers. And oftentimes, I think the first step is understanding it in the setting of just the natural history. That means you're not trying to treat it, but just does it exist? Does it differentiate a patient with FSHD from someone who doesn't have it? And also, maybe very importantly, among patients or people who have FSHD, does it differentiate severity of disease and discriminate across those patients? So I think that's a long-winded answer, but maybe to me, a very tangible, low-hanging fruit. And I think that is only going to serve this space well.
Thank you. And if you have additional things to add.
Well, maybe a reframing in the sense that the question was, how can the community get involved? How can individuals get involved, right? And I kind of think about it in three big buckets, which is basically trying to get to the same information, right? There's the piece of how do you engage in natural histories? How do you engage, and you address that very well throughout today, how do you engage with sponsors, right? To say, hey, what do we know? What can we share given our experience and expertise? And then how do we engage with regulators who are ultimately the ultimate arbiters of what happens and what needs to be demonstrated and what needs to be demonstrated? What should we measure, right? Which ultimately translates in what's most meaningful to the community, right? What can we measure? So there's what should we measure, what's meaningful. What can we measure, which basically speaks to the biology of the disease. What do we know about the biology? Again, being studied through things like natural history studies, right? And then it's how do we measure them? And that's the instruments, right? And do we have the right instruments that are going to measure what should be measured, right? And the discussion around instruments and design, is the conversation that sponsors have with the FDA, right? So they're only going to be able to respond and offer guidance as much as they know about your disease. So in educating them around what you're experiencing, what matters, how this disease is progressing, and what the state of the science is around the known biology, I think that's kind of how I think about the data that goes into designing an effective clinical development program.
Great. And then Eric, I think you can uniquely speak to the cohort C and how it's designed to take on these questions of how well validated is this novel biomarker, and how will C answer that question? What else will it answer?
Yeah, thanks. I totally agree. I think before I dive into cohort C, like the comments around, other biomarkers and MRI being really important, right? And then education, right? Educating regulators is really what's going to help drive that forward. And those are the foundational kind of discussions and comments that then allow us to move into some of these clinical trials where we can start to look at and validate some of these newly identified biomarkers, right? And so cohort C is that's the objective, right? So we'll be enrolling a larger number of patients, right? We're going to open up the enrollment to around for patients ages 16 to 70, right? We're going to enroll about 48 patients. And so that'll help us validate this novel biomarker. And, we need to look at it across like natural history studies too, right? So to your point, Wes, about looking at patients that haven't been treated and seeing how those biomarkers are moving. And really thinking about, do these biomarkers not only correlate to disease, but also to therapeutic efficacy? So we can start to measure that as well.
Another question is about validating MRI as a trial outcome measure. And the trials so far that have been run in FSHD have had whole body MRI, which is a challenge also, as maybe as big of a challenge as a biopsy, because they've gone for 45 minutes to an hour, right, each time they're measured. So I guess the question is, what kind of difference would it make to a trial if there were a validated MRI? And will we have a validated MRI? You can, I don't know, somebody want to raise their hand and start speaking?
Ladies first.
Katherine. Okay.
No, I'll just say, what difference would it make? I mean, for those of us who are in the process of designing the study, right? If there were a validated primary endpoint, I mean, that would be huge, right? And I think against current trials, it would be certainly shorter. Yeah. Probably fewer patients.
Why would it be shorter? MRI brings a lot of sensitivity that a lot of our other measurements don't. So I think MRI is hugely valuable because it allows you to detect changes even before you can see a functional change in a muscle.
Correct, right. So presumably, shorter, maybe fewer patients, right? So you can power your studies differently. Whether the cost of the study is different or the same, who knows, right? Because MRI is not cheap. But I think those are the three, and it would certainly minimize the burden on participants in a study. I think, all of that is qualified with, we have very little precedent for using an MRI, as a primary endpoint. So you would expect that you would be asked to also marry any kind of imaging with a functional endpoint.
The functional endpoint. And so I'll go back to cohort C and just ask you, so you're doing the biomarker, are you also doing MRI? Are you going to try to see how those relate to each other, and then the functional outcomes in C as well?
Yeah, so we will include MRI in that study. And I agree, the functional outcomes are important and they're really interesting, but they're so variable, right? Like they can vary from day to day. Like me coming off the plane this morning would change kind of how I would measure out on a functional scale. So having a standardized approach, to your point, right, not only helps us shorten and really direct our clinical trials, but then really makes a lot of advances in validating biomarkers like this, validating those functional endpoints.
Being able to draw a correlation between a novel biomarker that they have and something that's already well established is very important to the regulators. And so oftentimes you have complex trials that measure multiple things simultaneously, something that's well established and people know what to expect, and then something that's entirely novel, like this potential blood-borne biomarker that is really reflective of the disease, really reflective of DUX4 activity. And it's very exciting in that way. So, okay, Charlie, you had a question.
When would you release what this biomarker is?
So the question is, when might Avidity tell us?
As an academic, but
Yeah. And the reason I think that people ask is because they want to ask in their systems. They want to ask in their patients. And I think that turns it into a valuable marker even for patients and for Avidity, right? The more people that, the more data that is there, the more valuable it can become.
Yeah. Yeah, it's a great question. We get the question a lot. So,
Yeah.
and rightly so, right? Right now, our goal and effort is to really kind of understand if it is a reliable marker, right? And so as we run the study, get more information, then we'll disclose what the biomarker is. But I don't have a specific timeline for you yet, unfortunately.
All right, and Oliver has a question.
So you saw that CK showed the expected change, even a
Correct.
bigger change than your biomarker.
Mhm.
So yours presumably has the advantage that it's more directly linked to DUX4 expression. If that's so, is it the case that your biomarker does not show a change in Duchenne or myotonic?
So the question.
That is a great
Do
question.
you see that biomarker? And maybe you haven't
Yeah,
even asked yet.
Yeah, yeah, it's a great question. I don't know the answer to it yet.
Yeah.
So, but it's really interesting. I will think about that and
Yeah.
talk to some folks.
Yeah, because you would expect that if it's a DUX4 regulated protein, if it's a uniquely DUX4 regulated protein, it shouldn't be on and measurable in a Duchenne patient or in a Becker patient. But perhaps there are a couple of molecules that are DUX4 regulated, but are upregulated in other muscle diseases, and it could fall into that cohort. So I have a different question that, and I'll start with Wes and just move along, and it's kind of combining two questions, and if you could just, be very specific, but is there something unique about each of your therapeutic modalities that would indicate a specific patient type? And then is there something unique about your modality that would cause you to look for specific side effects?
Yeah, sure. So at a very high level, we are developing a potential treatment for FSHD that works by specifically and potently methylating the DUX4 region to shut down expression of DUX4. Okay, how are we getting that there? How are we getting that machinery there? We're using a vector or a modality that's called a vector, and it's a vector that's engineered and derived from a naturally occurring virus called adeno-associated virus. We're not new in that space, so that's not necessarily unique because, in fact, the type of AAV, there's different flavors of AAV virus. The specific flavor we're using is now in an approved treatment. So we didn't create this vector, but that's helpful to us because it gives us an idea of how well it crosses into muscle and penetrates muscle. And it also gives us some sense of side effects. So we know that AAV comes with a side effect profile that's becoming much clearer now that there's hundreds or thousands of patient years of experience with it. In addition to that, we have to think about potential side effects not related to the vector, but the actual medicine, right? That's working to methylate the region and then shut down DUX4. There's two things that we think about that so far in all of our animal testing and in our human muscle tissue testing that we've done in non-clinical like test tubes and those sorts of systems have looked okay, but we can't ever know until we put it into the clinic. So one is whether our machinery as a new protein that the body hasn't seen before would call in an immune response. Lucian mentioned this earlier. Again, so far that's looking good, but we have to be vigilant about that when we test this product in patients. And the second is whether or not the epigenetic editing is as specific as we want it to be and as it's been shown to be in all of our preclinical work. And that's something that we can monitor for in the clinic. So yeah, we expect that this drug could benefit all patients with FSHD. It's being designed as a one-time treatment. But all of these things, of course, you can't know until you test them in clinical studies.
Katherine, could you answer the same?
All right, so I'll qualify with Dyne is still in preclinical development, right? And so, uh, but we are using an approach that is under the kind of large umbrella of antibody drug conjugates, right? And our platform called the FORCE platform is really three components. We are using a fragment antibody, right, as opposed to a monoclonal antibody. We have a linker, and then we're using an siRNA approach as the payload on this, right? So three components to the drug with the idea that there's a more targeted approach to delivering to the cytoplasm of muscle cells, right? I think we are not, as I said, Dyne 302, which is our candidate molecule, is not in the clinic, but we do have clinical development programs in myotonic dystrophy type 1 and in Duchenne muscular dystrophy, right? Using the same Fab or fragment antibody and the same linker, right? So we do have a good safety profile in some of those later stage programs, which shows a favorable safety profile. So that is encouraging if and when we move into the clinic for FSHD. In terms of a specific patient type, I think that's part of, the process that we're going through right now is to say, what would that eligible participant look like? And as we lean on the patient community to kind of define some of these things, that's kind of we'll get back to you.
Yeah, it's a great question. I spoke to a little bit about the safety profile for Delbrex, and it's a similar safety profile we've seen across all of our AOCs, right? So the part that I'd like to kind of expand upon is the question about who's right for the trial, right? And so, we've gone in and we've really been thoughtful about trying to design our trial to answer the question of are these compounds safe and efficacious, and to be fair, in a subset of patients, right? So we're looking at patients that had moderate FSHD that were still ambulatory, right? It doesn't cover the scope of all FSHD patients, right? But it's part of the drug development process to start there and really think about safety and efficacy. And then it's really through collaborations with researchers, with the patient communities to really understand all of the different challenges and things that would be beneficial for our patients to then follow up these pivotal trials that are really designed to go to the FDA and regulators and get approval so we can get access for patients to these treatments. But that doesn't mean that the research stops, right? We keep going. There's lots of questions that need to be answered. And so we will continue to do research and open up different trials, similar to like our functional arm of our study, right? To kind of just grow the science and share it back with the community.
Can I just build on your answer for a
Sure.
second? Because you really sparked something that I think's really important, and you mentioned it as well, is, kind of deciding who or, kind of choosing who might be eligible is directly related to what do we know about the natural progression of this disease and prognostic markers of disease so that we are in a position to see change or benefit with an investigational drug. So that kind of choice in who participates is dependent upon the biology and what we can be testing through, outcome measures and the tools. So it's just complicated.
Yeah. And that leads to a question that's kind of asked in a different way, but what's needed in terms of research, policy, or advocacy that would allow the inclusion of pediatric or older patients or non-ambulatory patients who are currently excluded? And I think Avidity may be thinking about this most as your most advanced. So I'll ask you, Eric, to start.
Yeah, no, as I kind of alluded to in my earlier answer, right? Like, yes, we need to consider patients along the whole spectrum, right? And so we definitely are thinking about, how do we move forward after we've moved these registrational trials forward to start thinking about younger patients and early onset, to think about patients that have lost ambulation, right? And can we help those patients as well? And again, to your point, right? It's what do we know about the biology? What do we know about the symptomology, right? And so I encourage you to work with FSHD Society to really be a part and be vocal and help educate regulators, help educate us as we're involved, as we're embarking on these kind of journeys, right? And also, join and contribute to the natural history studies, right? Because that is really all of this learning is really feeds into an affinity loop, right? Where we understand one aspect, we go out, we test that aspect, we ask questions, we learn, and we use that to build upon it and really expand our research efforts and the patients that we can help.
All right, and I'll ask if there are any specific additional questions that this group has. Yeah.
Just wondering, and I know it's hard to answer because we don't know how everything's going to play out, but do you have any estimate of time for any of your drugs as to when they would be commercially available, like in past clinical trials?
I'll ask you to start first.
Sure, yeah. Yeah, it's a great question. I don't have a specific date for you. But what I can say is, one of the goals of cohort C, where we're looking at biomarkers, if that study turns out well, there's a potential regulatory pathway to accelerate approval. So we are looking at those regulatory strategies because it is our goal to really move these therapies along as rapidly as possible, while still ensuring that we understand safety and efficacy, right? But to really get them to patients as quickly as possible. So that's what we're trying to do with this biomarker cohort.
And there's been precedence in Duchenne's muscular dystrophy for accelerated approval based upon data from phase 1, phase 2 studies. What it allows is access to the new therapeutic while the phase 3 is ongoing. So they're anticipating starting a phase 3 sometime in 2025. It'll go for, let's just say, 2 years at the longest. I don't know any details there, but so that phase 3 could go forward. But if they have accelerated approval based upon the data they already have, well, they've showed us the 20 milligram data. They'll soon show, I imagine, the 40 milligram data, and then they'll have cohort C, which is the biomarker proof of concept validation study. And it's at the end of that body of work where they'll have about 130, 150 patients who have seen the medicine. They can take that to the FDA and ask for accelerated approval at that. And if it's given, the medicine can become available. And oftentimes companies are already planning for success based on their data, and they've even got the medicine produced by that point. I just am very aware of that. So other questions?
Because I think there's a two-part question. There's a two-part answer to that. There's a regulatory strategy to approval, which doesn't guarantee it's coming to you to take, right? And I think, it feeds off of the question that you just asked about, kind of what can the community do, right? There's a whole kind of body of work that needs to be done to address access to medicines, right? Whether they are approved through an accelerated approval process or a full approval. And that speaks to who are the other stakeholders that we really need to be educating, right? And that's the whole payer environment. What is the value of bringing this medicine to the market? Because remember, an approval by the FDA is really just an authorization to market. Doesn't mean anybody has to pay for it. So what is that body of work that the FSHD Society and all of us need to be doing to educate and to prime a system that is ready to pay for any medicines that are coming through an approval process?
Yeah. And that is active work that's ongoing by companies that are in the clinic and by the society and by other societies as well, in the United States, in Canada, and in Europe. That's a question.
People in the room would have FSHD too. Your clinical trials, including those. That was the question asked.
Yeah.
So,
will FSHD2 patients be included in the trials?
So yeah, we currently include both FSHD1 and FSHD2 patients. The one thing to keep in mind is that you need to have a genetic test to for diagnosis.
The epigenetic.
Yeah, and epics.
Yeah, so when we talk about who an investigational product is intended for, that's not necessarily the same for who are you initially studying it in. So while we think that our product can benefit both type 2 and type 1, or would have activity in type 2 and type 1, I should say, our initial study is going to only include type 1. That helps us to better make sure that the way in which the drug we think it's going to work is in fact working, so-called pharmacodynamics of the drug.
And the reason there, I'll just explain a little more, is that there's different reasons why there's hypomethylation of the FSHD, of the DUX4 site with FSHD1 and FSHD2, and they all have to address that question if both mechanisms are equally addressed by their approach.
In the case of rare diseases, and I don't know how, I don't have the numbers in my head, so I don't know how rare FSHD is. How does the commercials work? I mean, this is all very expensive. So, like, how expensive does a drug need to be to support this commercial process?
Okay, so he's saying in a rare setting like FSHD, where right now I think it's we expect 1 in 8,000 individuals to have FSHD in the places where it's been measured. How expensive is this? A challenging question. Does a drug have to be in order to be profitable or valuable to a company in a rare setting? You don't have to, yeah. I don't know who wants to take that.
I mean, I can just say, I think FSHD is rare, but it's not ultra rare, rare. I mean, of all of the rare diseases out there, I think it's relatively more prevalent. I think most estimates are at least 1 million people affected globally. It's an important consideration, no doubt. But I just wanted to make that observation.
It's really hard to name a number like that. You know, you look at where there's an ultra rare indication like SMN, and the numbers are huge, right? The cost of the medicine, as you said, FSHD doesn't fall there, but it's going to, the cost of goods is going to be important, the delivery of the new medication, as well as the number of individuals that you might be able to reach with the medicine. And it's a careful calculation. It's also got to be affordable to the organizations. So for instance, the European regulators haven't approved certain drugs because they have said that the benefit that is seen is not worth the cost that's being proposed by the company. And that's been different than what the FDA has said in the United States. So it's a really careful calculation within each company and perhaps within each region about how they set the costs. And it's very difficult to answer. There's a question there.
So along that affordability perspective, I wonder, I know Avidity, you mentioned that genetic testing is required, which can be cost prohibitive from an equity perspective. So I'm curious what your companies and organizations are thinking about to make sure that it's equitable participation, so you don't have too many barriers.
The society is working really hard to ensure that there's accessible and free testing for many individuals through systems that we're putting in place. I'll just start there, but you guys might want to have some answers also.
Either way.
Okay, so this is my passion project since my professional background is actually as a genetic counselor. Yeah, I think this is a huge area of concern and potentially an barrier to access to therapies, right? It's embedded in your trial currently. You've had sponsored testing programs under the umbrella of FSHD Society. I know that you're looking to expand that.
Right.
But I think there's also a healthy discussion from a policy perspective about, the value of earlier diagnoses, right? Getting to an earlier diagnosis that includes genetic testing and stops looking at genetic testing as just confirmatory, right? And I think that's where the cost comes in, because there's a gap in understanding that, clinical tests or clinical diagnosis, genetic testing is just going to confirm that, right? It's really about the access to precision medicine. And we really need to start having that conversation at a policy level, which hopefully would translate into greater access to testing and lower cost. But everybody else can.
Yeah. The technologies are also going to continue to evolve that lower the costs to testing. And so the evolution of those, there's already a very inexpensive test that's available, but it's not CLIA approved at this moment from saliva. And then there's other tests from blood that are much more expensive because they're much more technically challenging. And actually tell you more, so there's room for improvement. There is no question about it. And the society is actively engaged in trying to get us a less expensive and more broadly available test.
So one being made of natural history. And when you had that slide up, I didn't see anything in New England that was participate in the natural history.
Oh.
Is there someplace local? I mean, is this facility?
Larry, I'll let you answer that.
So the Clinical Trial Research Network was initiated by Jeff Statland and colleagues, and through gradually getting more and more support, they've added more and more centers to that. So I've been in discussions with them, and they certainly want to add UMass as a center, especially given that there aren't as many centers right in this local area. So that's in the works, and hopefully imminent, yeah, especially given all these upcoming.
And the closest one is Nick Johnson now in Virginia.
Rochester.
And Rochester.
So I just want to say, I'm one of the people that has hearing loss as an FSHDer. So if we could try to use microphones, it just helps me so I don't miss it all.
Would you like us to repeat the answer there, or did you?
I got
Okay,
most of it, but
okay,
if we can
great.
just try to.
Yes, we will.
Yes, I would just like to continue this discussion a little bit further for our panel. You have a Cinderella that's going to be the right fit for your clinical trials. I understand that the degree of severity, the age, the parameters in which you work within. The one accommodation I haven't been able to see yet, for example, just mentioned the closest place for a clinical trial is Rochester, which is a massive trip, depending where you are in this gorgeous section of the country. So one of the roadblocks I've heard over the years is time, cost, and availability. And some stories I've heard of, people have been in clinical trials and they only find out after the fact that there was reimbursement. So we have to consider the financial cost on the participant, because we are the boots on the ground. We are the people that you need and we want to participate. But there are many obstacles that stand in our way. Availability of where the trial is. I understand the complexity of setting up clinical trials. I get that. But we're a densely populated section of the country and we're a very active population. So we're ready. We just need some more help, some ease of accessibility into the trial once all the parameters for that trial have been agreed upon between clinical people and the participants. So that often will stand in the way.
Yeah, so it's an important consideration for ease of accessibility to being a participant in a clinical trial. And the challenge with the clinical trials for FSHD is that they have been complex. They've included MRI, and it required there were specific MRI requirements that were available at limited number of sites. That was initially, and now we have some exciting data from Avidity that could allow us to expand that out, where perhaps we won't have to have MRI going forward. Perhaps there'll be other ways of measuring the disease precisely that should ease access. The society is taking this on in that we've heard from patients that it's very hard to get to some of these sites because they aren't close by. You've got to fly, you've got to drive, and also they're not taking new patients. That's the other challenge, is that the sites become very crowded, especially as the number of clinical potential therapeutics has been increasing. And so our goal is to identify sites in Canada and around the country that act as satellite sites that allow for treatment, allow for therapeutic evaluation, but don't have all of the huge requirements that you might need in a first visit and a last visit. So there is active work ongoing in that question, and we hear very clearly it's an issue. You're very welcome.
I'm a little bit confused. So is this site then not being looked at for the trials for some reason? There's something missing here in
No, nothing missing here. This, yeah.
Each clinical trial.
Wait.
Yeah, I can share up here, so just take a hand, I'll go after.
So as we've heard, each clinical trial is it's an extremely complex system that has to be set up and put in place. And to Fulcrum's credit for pioneering in this area, you know, at UMass, we've participated in their two trials, their two treatment trials, and that involved getting the MRI clearance. There's all sorts of other factors that companies look at to see if we would be an appropriate site. And we've been in discussion with others in this group here. It still does take longer than I would like to get the IRB and institutional approval, things like that. And so one question I have is whether companies can, you know, and the FSHD Society can work on ways to streamline this process so that other sites, you know, not just ours, but other sites that might be in sort of desert areas of clinical care could also be involved so that people aren't left out. And, you know, because I think the more we get experience with this, the more there's evidence from other areas of sort of unified IRB protocols and things like that that can really speed things up. So I'm really hopeful, but we are already reaching out to try to be a site here as well for future trials.
Future trials, and they were involved in the first series of trials from Fulcrum. And I'll just say it one more time, the society is working very hard, and that's one of our primary goals for the next year is to bring additional centers throughout the US and Canada that can carry out these kinds of studies. Go ahead, Katherine.
I also want to address the financial burden of participation in clinical trials that I think it's Kathy, right, that you brought up. And I know that the society works very collaboratively with other advocacy groups and other disease spaces, but, you know, I will say that, you know, in some communities that have had decades of experience in clinical trials, whether they've been successful or not, have put out toolkits on this very issue in order to harmonize across sponsors what happens in clinical trials, in particular, against financial burden, whether that is travel, you know, do you use prepaid debit cards, do you use a concierge travel service? So all of this is mapped out in PPMD's clinical trial toolkit, right, which might be a really nice tool for this community. Of course, it needs to be tweaked when we think about, you know, what are maybe some challenges to a pediatric population participation as opposed to an adult, which may have, you know, many more issues around accessibility and you know, things like that. But I think there are models that are out there to reduce the financial burden on folks who are considering and participating in clinical trials.
And then I want to ask the question around access to pediatric or elder patients or non-ambulatory patients in a different way. So for instance, a drug achieves accelerated approval. Who can receive it under the accelerated approval? Because FSHD seems to me to be a unique disease with a knowledge that DUX4 drives it. Individuals who are pediatric or over 70 or non-ambulatory for sure, the disease is being driven by DUX4. So what's the calculation that's going to be once you achieve accelerated approval, who actually has access?
Yeah, it's a good question, and I think it's going to be a two-parter for me. You know, part of it will depend on our discussions with the FDA, right, and the regulatory bodies, and what our prescribing information looks like, right, and that's the regulatory hurdle. But to your point earlier, right, the next hurdle is with payers, right, and they're going to evaluate all of the data that we have, and they're going to make a determination of who they're going to cover, right, and so that is work that we're thinking about up front as well, right, because it's not just one hurdle that we have to get over, right, to ensure that folks have access to treatments, right? It's kind of a two-pronged approach, but we are aware of those things. And so those conversations and those work, the thoughts and ways that we approach that, we initiate early on, right? So we can start to get a better idea of who may be able to get access through an accelerated approval program or just a regular approval program. It's not just unique to accelerated approval pathways.
On that same type of topic, and it doesn't necessarily have to be towards your company in general, but just drawing off of your experience in the industry. Why is it that severe patients in rare disease are left out? And I've been to 2 roundtables in the past 6 months with the FDA where they're saying they want to see cohorts that are across the board in severity with rare disease, but you're not seeing that activated in any rare disease space, whether it's FSHD or other muscle diseases where the severe patients are being addressed. The Cinderella, back to Kathy's point like that, the Cinderella patient is still the one the sponsors are picking, even though the FDA is open to and actually encouraging a little bit more creativity on the clinical trial design.
I just want to bring up an important point and an important observation. I think for FSHD, given the clinical heterogeneity, when we talk about the Cinderella patient in a clinical trial, that's not necessarily, I think, the patient that drug companies and academics are trying to develop treatment for. We're trying to develop a treatment that helps all patients. But I'm going to bang this drum of a biomarker again, because a surrogate endpoint, a biomarker that serves as a surrogate endpoint doesn't just get patients an effective drug faster. It also then becomes an entree once you've proven it in the full clinical study, right? That you've made the correlation between this novel surrogate endpoint and a way that patients feel, function, or survive. Once you've made that correlation, now you can start to study it in more difficult to study populations from just a classic functional endpoint perspective. And if you can show that in those more difficult populations, you're moving the needle on that biomarker just like you moved it on that initial Cinderella patient, which was, or population, which was the population that was used to prove the correlation, right? Because to prove the correlation, you have to be able to predict how those patients are going to progress without the therapy. And there has to be proof that there's enough difference. So I think of it as, again, the importance of these biomarkers that can serve as surrogate endpoints are not just getting an approved product to that Cinderella population faster, but then allowing us to get an effective medicine into patients that are more difficult to prove that in because they're so heterogeneous. So
Mark, in a patient who's also
if it's
months versus 1 year.
Yeah, if it's a universal biomarker. So if it's a universal biomarker that it makes sense in the pathway of the disease, that it should be sensitive and specific to read out the disease and an effective therapy, and we can measure it readily in people, then yes.
And imagine it was MRI, or imagine it was a circulating biomarker and we've got that data that shows that it's a good surrogate for the disease. It becomes very valuable in this case. And the other thing I will say, and we're 10 minutes over, so I think after this, we might just call it an evening, but the Fulcrum study, I'm not arguing that all I'll say is that the placebo arm didn't progress in the way that they anticipated, so they had nothing to compare the treated arm to. And the reason you go for subjects, I'm just reiterating what Wes said and what we've spoken about, the inclusion criteria in something like FSHD is intended to minimize the variability and so that you can predict progression in your placebo arm. It has to progress enough that you can compare your drug to, and the reason, one of the reasons that the Fulcrum drug did not show benefit is the placebo did not progress. They had accepted a wider range of functionality in the patients in their phase 3. So that's the challenge to us in a disease like this. You could miss the benefit. If that drug were going to be beneficial, it could have been missed because of that failure to progress in the placebo. So that's the challenge to us as drug makers, is having to minimize the variability, get the progression in 12 to 18 months, be able to compare in the placebo, be able to compare that to the treated, be able to say it's efficacious, have a biomarker that is a surrogate for the disease and be able to take that to the regulators and payers. And that is clearly what we're doing. And we're not there yet, but that's the goal. Okay, I will take one more question. I can't end with me commenting.
This question I have is, for people who are treating their disease with physical therapy, exercise, actively actually growing their muscle and those cases, what type of problem does that cause for judging of efficacy when those people are also growing, treating their muscles with other methods?
So the recommendation, the question is if people are actively exercising and doing physical therapy and things to benefit themselves and maintain their strength, does that cause a challenge? What Fulcrum said, was just continue what you were doing. Don't change what you were doing. And I imagine that they're all saying the same thing. There have been small clinical trials that show that physical therapy and physical activity to a certain extent are beneficial to FSHD subjects. And actually another exercise study is being done by Jeff Statlin and the CTRN to demonstrate that in a larger population. So if you've got something that works for you, we don't want you to stop. We want you to continue with it and continue it all through the trial.
Is it a risk though that it's going to cause disability, the only therapeutic that's currently trial?
I don't believe that they're the only therapeutic currently in trial. Perhaps in the US?
Is the Roche study open?
Well, the Roche study is open.
Yeah.
It's fully,
Okay.
no, it's fully enrolled. So there's not taking new patients.
It's in the Netherlands.
Arrowhead's in the Netherlands, and then there are folks in Australia. So maybe in the US currently it's this phase, it's this C cohort. All right, I want to thank you so much for your active participation. I want to thank our members of the panel for coming and answering some really good questions. And we'll do this again sometime.
Stuff done.
Thank you, everyone, for coming. I think this has been a fantastic educational day, and we've covered the whole range from some of the science to some of the ideas about inclusiveness, including the pediatric ages, and hearing about some of the complexities in designing clinical trials. And so I thank all the participants, and thank you for all of you who donated blood samples. And just thanks for taking your time out of your day to come visit us here. Thank you.